So next is Herbert Sauro from the University of Washington. Herbert has thought about modularity a lot, has published on it, and has his own point of view.
I do. So first of all, I want to say thank you for this this very nice location, by the way. And the hotel is so close to this, so which is very nice. So I'm going to talk about. I'm going to give you maybe controversial. Well, I'll try to be controversial to make it interesting, since this is a workshop, on a perspective that a number of us have been thinking about. And of course, I attend Reinhardt's modularity meeting every 2 weeks, and we also have our own sub meetings at UW as well. And the individuals in the sub meetings is Steve Andrews is in the front, and Joe Hellestein, both of those at UW in Seattle, and then Song Feng over there, and Steve Wiley at PNL. So we meet quite regularly as well to talk about this idea of modularity. And I'm going to, I thought I'd start, actually I'm going to skip right to the end here. I'm going to briefly mention how I got into this. And also because it was mentioned this morning that the NSF used to have a theory in biology program, and they don't have it anymore. And I keep telling them, why don't you reinstate it, because it was so useful. And this bit of work actually got me into modularity and was funded by that theory program from the NSF. The beauty of that program was you could just do theory. You didn't have to attach experiments to it, so you could focus on conceptual problems. And the proposal I had was whether we could use genetic algorithms to evolve networks with specific functionalities. Right, so these are chemical networks. So I'm a hardcore systems biologist. I've been for many, many, many years. And I thought, could we evolve specific functionality of using genetic algorithms? And the quick answer is yes, you can. You can pretty much evolve whatever you want. And the first thing we thought we'd try was, can we get a network to compute a square root? And that was too easy, it turned out, because it turned up with this thing. So this little network, and I guess if you think about it enough, you'll think, yeah, of course, this probably can compute the square root. The input is x, right? And the output, which is node 3, actually turns out to be the square root. Precise, exact, okay? And it turns out you can evolve lots of them. These all compute square roots. And so we thought, well, let's be a bit more adventurous and compute, say, solving a quadratic equation. Yes, it solves quadratic equations. That's really nice. Sort of keep this structure in mind. And then we thought, how about computing cube roots? And to my surprise, yes, you can compute cube roots. But what was really surprising, after I rearranged this network, there was a quadratic equation solver right in the middle. And of course, this got me thinking, what's going on here? So there's some algorithm here it's implementing. It might be the old 16th century algorithm for computing cube roots, but it involves evaluating a quadratic equation. And you can see there's some division of labor here, and there's this thing in the middle which computes which solves the quadratic equation. Yeah.
So what do the arrows mean here?
Oh, sorry, I'm assuming too much. So I'm a systems biologist. I study everything inside cells, right? That's my, yeah, sorry, you're right. That's my thing. And inside cells are full of chemical reactions, right? Bindings, unbindings, degradations, transformations, all sorts of things going on, involving small molecules, proteins, nucleotides, etc. And these arrows represent those chemical reactions. So if I go back to a simpler one, for example, this reaction here from 4 down to 2 is a unimolecular reaction that degrades species 4 into some waste product. Okay, and these are simple mass action kinetics. Yeah, I should have said this. These are simple mass action kinetics. So the rate law here is some rate constant times the concentration of 4. This one here is a bimolecular reaction where 3 combines with 4 to form 4. Okay, where in a sense 4 is acting as a catalyst here because it neither gets created nor destroyed netwise. This one is a splitting of 3 into 3 itself and 4. Okay, that's what these are. Yes.
So you showed us the multiple different ways how you can compute the square root. You showed us one way to compute the cube root. in any networks that give you the cube root, do you see the square root one, or is it?
This is a very good question. Unfortunately, it was 20 years ago, and I can't remember. We got a couple of cube root calculators, and this is the one that stood out in my mind, and it's always been, I've had a slide of it for a long time because of that. But yeah.
Why are there multiple self interactions?
Multiple, what, like
two with itself and then with itself again.
Okay, yeah, that's part of the evolution. So the evolution allows, so the evolution algorithm works like this. So we generate 100 random networks. Those networks can contain uni-uni reactions, uni-bi, bi-uni, bi-bi. We do not restrict what the products or reactants can be. So sometimes you'll get A plus A goes to B. Sometimes you'll get A plus B goes to A. So it allows all those possible combinations. Then from generation to generation, we compute a fitness function, which is the target function we want. And in each generation, we will keep the top 10%. They go on to the next generation. So we rank them based on the fitness. We take the top 10%, those are the best. The rest of them we mutate by either adding reactions, deleting reactions, or mutating the rate constants. All right, and then we use something like roulette, tournament selection, actually, to select the remaining 80 that we need to recover the next 100 in the generation. Then we do the ranking again, and we do that. This takes about 100 generations or more, and you'll get the function. All right. It's a fun game because you're there watching the fitness function. Is it going to go down? Is it going to go down? And you can do this all day long because sometimes it never makes it. Other times it does, right? So and after a while, you realize, I know this one is going to converge because I recognize the way the fitness function is dropping, okay? And
just a question about, let's say once you actually do have this particular configuration that you've arrived at, is this X something that is continuously being replenished? And does this
so I should
steady state, or what is
it? So the magenta ones here are fixed species. They're boundary species. We fix them.
I see.
They don't change over time. All the others, the black ones and the green ones, do change in time. The green one is highlighted because that's actually the answer.
And that's like a steady state you get to at the end.
In this case, it's steady state. But we also do time-dependent ones. For example, evolving oscillators is very easy. In fact, we have on GitHub, there's about 200,000 oscillators that we generated. We can also generate bistable switches. We can generate bandpass filters, low-pass filters, high-pass filters, things that resist perturbations. You pretty much can evolve anything you want, really. We have code in Julia and other languages. You can run them on clusters to speed things up and so on. You can generate large numbers of networks. Okay, so that was the surprise there, and that started me on the road to modularity, basically. Okay, I had no idea what modularity meant at that point. I just saw this blob in the middle, and that looks interesting. Should I use the word module or not? I don't know. So now I can go back to the beginning now. So I've been thinking about this on and off for quite some years. And from the discussions, there are a lot of opinions on what a module is. Okay, it's a highly overused word. So let me start with some terminology, things that are not, what I think are not modules. Okay, so a simple mechanism like those evolved networks. I'm going to call them subsystems for want of a better word. Okay. And they just carry out some function like an oscillation or a calculation or something. So two or more biochemical processes forming a network whose species evolve in time. An example of those are the ones by John Tyson. So some people so this paper, John Tyson and Chen and Novak, Novak actually passed away last year, those of you who don't know. He was at Oxford. He was a computational biologist. So that was sad to hear that. But they published this famous paper now in Current Opinion in Cell Biology, titled Sniffers, Buzzers, Toggles, and Blinkers. That's probably why it became quite famous. And in there, they have a whole bunch of networks. And I consider these to be subnetworks that carry out some function. I don't know if they're modules or not, but they certainly, because we haven't defined what a module is yet, but they certainly do stuff. Okay? They do stuff, all right? And those are good examples of such things. Now, depending on the definition of module, they may turn out to be modules, but they're definitely networks that do things. Okay. The other one, which we'll probably hear about from Yuri Alon later, is this idea of a motif. At the extreme definition of a motif is simply a topological pattern that you find enriched in some graph network. That's it. Now, of course, Yuri Alon went further, and he actually instantiated these patterns as feed-forward networks and then studied their kinetic properties. All right. So motifs are sort of, they're just patterns. They're sort of dead. They don't do anything, but they are enriched patterns in a much larger network. And then we have a module, and so to be discussed, because there are lots of interpretations of that. If you go to Wikipedia, there are at least 18 sub-definitions of modules, and in each of those, there are probably other variants too. There are large numbers of interpretations of what this module is. So I just playing around with Google Slides, but so this is the way I defined it. So subsystems is a large group. This is one of John Tyson's. This is, I think, is a relaxation oscillator. Motifs. Motifs are maybe another group. They're sort of topological, and then we have a small group that might be modules, but we don't know what they are. But they overlap with motifs and subsystems, assuming you actually instantiate a motif as something that can be implemented in a computer that does something. So that's sort of the general terminology. So since this word is so overused, what is its origins? Well, it actually goes back a long way, actually back to the 15th century, I think, but it meant something completely different then. It meant to measure. And it was only in the 50s that it became more widespread. We found more widespread use of it in engineering, and it was picked up by the post-war electronics industry and the emergence of computers. It entered into the dictionary, I think, in 1955 as an interchangeable part. Right, that is the modern origins of the word module. NASA also picked it up and called their lunar spaceship a lunar module. Okay. By the way, one little side note. So we just saw the Artemis mission. I don't believe they used any new math to get to the moon. It was Newtonian math and perhaps 1960s control theory math. So they didn't need any new math, just in case we need new math. But anyway, so this is an example of a module. If you go to eBay, you can buy these now for about $40, $50 from the 50s and 60s. This is a PDP-8 transistor diode logic module. It's literally called that. It's a triple flip-flop from 1965. These were cards that you could plug in and pull out. They're the little handles. These are very specific functions. And so then if the computer broke down, an engineer would go in, they'd find the card that was faulty, pull the card out, put the new one in, computer would work again. Okay? So that's why it was an interchangeable part. If you look at the evolution on some trends in books, at least, you see it rising, did nothing pretty much for the first 1,000 years or so, and then suddenly in the 1950s it just shot up, and it seems to have stabilized a bit in recent years. Right, so this is a very new word actually that's entered our lexicon in science, which is why it's probably so ill-defined everywhere. So although the electronics people say it's not ill-defined. Where's the gentleman who's speaking up about electronics? There, yes. And that is exactly the point. So then there was this very famous book by Hartwell, et al. In 1999 on from molecular to modular cell biology. And I guess that was the paper that brought the word into biology. Some of you may have known other references before that of the use of the word, but not the idea, at least the use of the word. And they emphasized, I read the paper again on the plane, and I realized they don't really define it. It's not a very interesting definition, actually, what they gave. They called it a functional module. Is by definition a discrete entity whose function is separable from those of other modules. Now, they don't discuss what it means by separable. So, I mean, the essay is really interesting, of course, nothing wrong with the essay. but ultimately they didn't really pin it down in that paper. But anyway, since then the word has dramatically increased, and I just went to PubMed to search for the word module, and it's got about 80,000 matches. So and there's only 50 of us here. So where are the other 100,000 people? Okay, so now on the other hand, so we have one interpretation of a module there, some functional module. That sounds a bit like those Tyson models. But on the other hand, if you go to the esteemed Encyclopedia of Systems Biology, the book chapter on modules defines it as a biological module is a subnet entity composed of nodes that are more closely related among them than to the rest of the components in the networks. And that, of course, was all the rage in the 2000s. The Barabasi, I don't know what to call it really, but there was a lot of excitement over graph models in the 2000s. I was looking at it and thinking, yeah, academically and scholarly, it's wonderful, but I'm not sure what it tells me at the end of the day. And I did look into it quite a bit. I really liked it actually as a scholarly endeavor. It was really nice. But in the end, I'm not sure how much it contributed to biology. But if you're interested, the two papers which I really like. So first of all, that diagram there sort of depicts this idea of a community where you have highly connected nodes separated by a fewer connections. And therefore, these highly connected nodes are therefore considered modules. Right? That's a fair definition of a module. Not complaining about that. And that there's a nice paper in PNAS that talks about that. There's an algorithm to compute these clusters, and there are lots of software now in Python and so on that'll compute these clusters for you and identify them if you need to. Those of you who are interested in a really nice review of that era, I call it that era back in the 2000s. Newman in particular had a really nice review. It's on arXiv, but turned into a SIAM review. It's a really nice review that basically covers the field. Nothing wrong with the field itself, but in biology, I don't know what it ultimately contributed. There was a paper in Science that came out by a well-known group. They did this on metabolism, and they found that things like pyruvate, glutamate were hubs. Therefore, they were important. If we knocked those out, we could disrupt the hub in the metabolic networks. But unfortunately, you cannot do that to metabolites. So I wrote to them, but they never replied, letting them know you can't actually do that, by the way. But interesting paper. So now it's used lots of other places. For me, I'm actually a biochemist, microbiologist, but I have a master's in computing and computer science. So I'm aware of some things that were called modules, like operons, in bacteria in particular, called operons. And to go back further, back to the 50s, the 1957 book, does anybody know this book? Probably you do, Waddington, Strategy of the Genes. He talks about developmental pathways. I'll have a slide on that. I'll talk about that in a second. But at Caltech, they did a lot of work on sea urchins, and they noticed there were conserved transcriptional networks across species of sea urchins, and they were termed modules as well, if I remember right. And then protein complexes, the big things like ribosomes, or the smaller things like pyruvate dehydrogenase have often been called modules as well. And of course, back to the protein interaction maps where you see high clustering as networks. And then also people will call the Tyson networks modules. And then pretty much anything in synthetic biology is called a module. And so I did about 8 years of synthetic biology, and then I realized there's no grants in it, so I had to switch back to systems biology. But pretty much anything in synthetic biology is a module. So now I love AI, by the way. So you'll see a couple of these just for fun. What's the general consensus? I think the general consensus perhaps is the word module seems to be attached to anything that is a collection of closely connected parts. All right, be it genes, network graphs, etc. Now, is that a sufficient definition? I guess that's the question for this week. Okay, and there's a beautiful rendition from Copilot. I don't normally use Copilot because it's not very bright, but it's not bad at creating diagrams. And here it depicts the module and all the different things around it. I told it, I don't like all that fluff in the background. Could you get rid of it? And it did actually, but I didn't show you that one. So Waddington, those of you know, he was an early developmental biologist. He had this thing about developmental pathways, and he had this idea that there were these sort of unseen puppeteers underneath this landscape, which were basically the genes that were orchestrating the shape of this landscape to direct organisms in different directions. And they were essentially the modules. You didn't call them that, of course, but that's basically how you might interpret them today. And because Waddington and people at Edinburgh, they were way ahead of their time in the 50s. And there's a little known appendix to this book by Henrik Kutcher. This is probably one of the earliest systems biology papers you'll find, some physicochemical aspects of biological organization, which is an interesting paper. Probably the earliest systems biology paper, for those who are interested in history, is probably 1934 from Sewell Wright, who postulated the idea that genes were expressing proteins that made pathways. And at that time, genes were barely understood, other than as an abstract thing. And pathways, well, sort of glycolysis was known, and so that was really a remarkable paper, really, at the time. Okay, let's go back to the 1950s then, back to the engineers. So if you asked an engineer in the 1950s, what would you list as the basic properties of one of your modules? The first two, they would probably say, well, there's a defined interface here. Okay, I can plug it in. So there's a defined interface, and there's definitely a defined function, maybe more than one function. There's a defined function, but they would also say this. A lot of text here, but it's something the gentleman, where is he again? There. Sorry, I don't know your name. Mentioned.
Bob.
Bob. That's an easy name to remember. He said, well, the output has to have a low output impedance, and the input has to have a high input impedance. And the reason for that is you don't want downstream modules to affect the functioning of the upstream module, right? And you don't want the upstream module to pull too much from, you don't want this module then to pull too much from upstream modules in terms of current to affect them. The idea is that the function of a module remains coherent even in the face of having to interact with other modules. That's what the engineers wanted. This is essentially what made engineering possible. The fact that you can take a thing off the shelf and it'll work within limits, of course, in whatever context you put it in. Right? So you mentioned context this morning. These modules the context is important because they exist within a context, right? And they have to maintain their integrity within that context. Okay, and digital. Those of you, I grew up in the 70s as a teenager, building digital electronics and computers and so on. Those of you know the 740 series. Those are built with this in mind, right? You can hook up. So the 740 series is basically a large range of chips, little chips like $1 each. Now they contain logic gates and so on. You can hook them up in any way you want, and the whole thing will work, guaranteed. Okay. There are limits. For example, a particular gate can only drive up to 10 gates. It's called a fan out. And we wrote a paper again on the theory of biology grant. Another one, I got 2 of them, lucky me. On what was the fan out for a transcriptional regulator. Okay, so the same, because transcriptional regulator can only control so many operator sites. After that, it sort of fails because there's not enough of them around, and whatever signal it's got is so degraded now that the downstream genes can't work. Okay, so there's that. So if we summarize then what the engineer might say, they say, well, it has a well-defined interface, meaning the interface defines some boundary. Now, of course, in biology, the boundary is not so fixed like in electronics, which is hardware and everything is distributed in space. Transistors can't move about to another part of the circuit. They are fixed, whereas in biology, of course, proteins and so on, they just float about all over the place. So you have a liquid state, so the interface might be a bit fluid, but there could be a well-defined, but at least in electronics, there's a well-defined function with an input-output behavior, and they are composable. This is this business about I can connect things together and they won't mess each other up. So now these are nice definitions, which can be almost made within the operational characteristics of electronics. These can be made almost perfect. Now, biology is a lot more messy, and we go back to this argument. Yes, you might have a beautiful theory, but does it work? It doesn't matter because it provides a framework on which you can use to talk about the biology, even if it's imperfect. Okay. So yeah, composability is the guarantee that when modules are assembled, they function. So of course, I asked Claude, draw me a nice picture of this, and he sort of drew this one. Yes, Claude is my favorite AI. Copilot is my most unfavorite one. But anyway, it's sort of try to depict these ideas: interfaces, functions, composability. Okay. All right. Probably coming to the end because I want to talk about more about discussion. Somebody mentioned this morning about modules of modules. This is also the only reason why this laptop can be built. I mean, the modern chips, and this is a fairly old laptop, but in modern CPUs, we have billions and billions of transistors. How on earth do you comprehend that as a human, right? We don't. We work in a hierarchy of modularity, and each level of modularity is described in a different way. So in electronics, and I talked to Intel people about this, so I haven't made this up. You can start with the lowest level, which is the transistor, which is physical. You can then build from transistors gates. So these two are roughly the same method. You use the same math to describe these, but then you jump to systems of gates, and now you switch to Boolean, which is a completely different way of describing these modules. So this is a half adder, I think. And then from this, you'll construct a full arithmetic unit, again, Boolean. But then you'll put lots of these things together to form a flow map, showing you how all these super modules work together. And finally, you have the chip itself. So the electronics industry has to do it this way because the level of complexity is just enormous. And by the way, they do not simulate single chips in their entirety because they cannot do it. You cannot simulate, 4 billion transistors, as a set of differential equations. That's what they tell me. And so they have to divide it up like this. So each stage has its own mode of simulation. All right. And so this is basically how they do it. Okay. One person I should mention is Del Vecchio. Unfortunately, she can't be with us here. She has taken this approach to a long way. She's at MIT. She's a control engineer with an interest in biology. I think we're going to get a talk later this week from somebody from her lab. The paper's not easy to read, but there's a lot of interesting stuff in her work. So we should bear that in mind. And then one last thing I want to talk about is, okay, so we have this idea of modularity. How do we approach it mathematically? So I listed some things here. I didn't list Boolean logic, but here are some ways in which we can use mathematics to study these systems. Of course, the old chestnut, which is dynamical system theory, which probably you all know. These are all work on differential equations. So dynamical system theory, correct me if I'm wrong, but basically focuses on stability, bifurcation analysis, chaotic dynamics, and so on. And so my co-pilot also made this also beautiful figure here. And of course, you notice the background is gone because I told it to get rid of all that fluff. It promptly did it. But here we have, bifurcations and so on. Basically studies, that's his focus, I think. And then we have these other areas like Martin Feinberg's reaction network theory. I don't know how many people know that. Bruce Clark's stoichiometric network analysis. These are very nice mathematical approaches. They basically focus also on stability, but based on topology alone, right? So just given topology, what can you tell me about the system? Is it stable or not? And then we have what the engineers brought in, which is control theory. It's not a well-known area outside engineering, I get the feeling. In fact, there are special reviews for physicists to try to introduce them to this field, if you look in the literature. But I particularly like this approach because it goes beyond just stability, because it talks about robustness. It talks about, in particular, the role of feedback and all the advantages of feedback, not just in terms of stability. For the engineers, of course, it's used primarily for design. And biology, you can use it for reverse engineering. So for me personally, this is the approach I like because it covers a lot of the bases that I need anyway. There may be other approaches here that I haven't listed, okay, including what we heard this morning. And so this is, so I'm going to do later on this week, we can have one of the work groups is going to be an introduction to this, those of you who haven't come across this, but it can tackle feedback patterns, robustness, stability. And what's really nice as a biologist, it lets you relate the system behavior to the component parts. And I was in the genetics department for some years, and the obsession was, can we describe phenotype from genotype, right? That was the obsession, and it's a good obsession to have. And it helps you address part of that at least. And this is just to say we'll probably have some tutorials and we'll have the workshops later in the week. Another beautiful diagram from Copilot. And then finally, acknowledgements. Then I'd like to acknowledge some years ago, I got 2 of these theory in biology grants. I thought they were great. I'm so sad they discontinued it. The first one was this evolution work, and the second one was trying to define whether we could apply control theory to stochastic systems at the gene regulatory level. And there we talked about the fan out for a gene. So if a gene controls multiple downstream genes, for example, how many genes can it control before everything falls apart? Okay, so that was the effort there. And then we have the Reinhardt meetings, which are great. And then we had also our own discussions with Steve Andrews, Song, Joe, and Steve Wiley as well. So those are great. All right, so I think that's it. There's some resources here. A nice book is from Brian Ingalls. I shared an office with him many years ago. It was a fantastic opportunity to get to know control theory. He taught me a lot. He's now at Waterloo. He wrote a really nice book on mathematical modeling. He's able to bridge the 2 camps between dynamical systems theory and control theory in a very nice way. And he's a fantastic writer. And he wrote this book basically that bridged biology with control theory, which is nice. And I think that's it. Yes, thanks very much. Yes. Maybe somebody could pass around the microphone. Yeah. Unless you can shout, of course.
Yeah, I can shout.
Yes.
So what's the answer? How many genes can a transcription factor
I think it's about 10 to 20.
10 to 20?
Yeah, but it depends on the concentration of the transcription factor and the turnover rates and things. But
I feel like they're like master regulators that control more than that.
Yeah, and they must be doing something special. One special thing you can do is add a negative feedback on the transcription regulator, so it's always stabilized, even though it's been drawn down by binding from downstream operator sites. So that's one of the things you learn in control theory. If you want to stabilize something, wrap a negative feedback around it.
Okay.
Yeah.
I can also yell. So
nice.
I want to get back to this idea of the motif versus the module.
Yeah.
Would you consider a feedback loop consider a motif or a module?
Well, from my perspective, it depends on whether it's composable. That is one of my criteria. Now, I'm not saying there are modules in biology, right? This is a hypothesis, okay? But are there these kinds of things in biology that are composable?
Let's say a feedback loop in a.
In fact, feedback loops help you compose actually, because they stabilize the endpoint. Yes. Okay.
It is actually a little bit hard to hear.
Yes.
So it would be helpful to have the microphone.
Okay. Good.
Sorry.
Yeah, these walls seem to absorb sound, I think. Yeah.
No, I can.
Yeah.
Building on that earlier question, over there, was that based on empirical results or
Which what?
the
Fine now?
Is it 20?
Well, there's a whole lot of theory, and then we tried to take numbers from the literature and see what it meant.
I see.
Yeah.
Okay, that's it.
So the whole thing was based on a Bode plot, actually, where the Bode plot with the bandwidth of the Bode. I don't know if you know Bode plots, but they basically plot the amplitude of the signal versus the frequency. And there's a thing called the cutoff point, where as you increase the frequency, the amplitude just dives down exponentially. And that bandwidth is basically the range over which you can drive downstream operator sites. So if you exceed that, you fail.
So are there any known biological examples of the things that you were showing, that, you know, networks which can do square root calculations and the Hilbert calculations? And
probably some bacteria in some vent down in the Pacific Ocean, maybe you can do it. I don't know, but no.
It's
Probably
at least from your side, it's not known.
I know it's not known. No, nobody knows. This is just fun, right? That was a nice thing about that program, Theory in Biology. You could do whatever you wanted. And you said, can we compute square roots using biochemical networks? Yes. So what?
Yeah.
Well, so what? Because it stumbled on that quadratic equation subnetwork within the cubic calculation. Yeah.
So for composability, like, is the main idea that like the eventual behavior should be somehow just a composition of parts, or do we also have some restrictions on like how fast decays happen, for example, to the steady state? Like,
say it again, the last bit.
So is composability mainly the thing about like what is going to eventually happen once I put things together?
Yeah. So if I have two things, right? Actually, I can show you. I can show you a demo. What about my demo? So here, so what I have here, I'll show you what happens when something's not composable, right? So this little network is actually one of Tyson's relaxation oscillators, and there you see it's ticking away nicely. I'm now going to connect it to a downstream unit, and I'm going to control the amount that unit draws the signal from the relaxation oscillator. Okay, as I do that, fail. If I was so bad, it stopped oscillating completely. And so the idea is, if this is not composable at this level, it is composable down here. Oops, down here. But it's still affecting the amplitude and the frequency, though. So it's not that great either. So this Tyson model, this subnetwork, is really not a module in my world, because it's actually quite sensitive. So if this was a clock, right, in a cell, and there are many clocks in cells, and you depended on this clock having a specific frequency and amplitude, and you didn't want it to change, and this clock had to drive something else, but it has to be designed so that when that thing starts using up one of the species in the clock doesn't change its behavior. Now, I don't know whether these things exist, though, in nature, but it would make sense if they did. Okay. So I'm not saying these modules exist in nature, although we probably have one example at least. I don't know how they're widespread or not, and whether evolution has just mushed everything together and just hope everything works. Or now, Yuri Allen is going to give a talk, I know, on modularity, the emergence of modularity in varying environments. So this is a paper you did years ago, which is rather interesting. And it says, so evolution may be pushing systems into this composable form, but it's not an answered question, really.
Right. I have another question. So thank you very much for the presentation. I really like the various components of modularity in biology. One side of biological theory that I didn't hear you mention that I think is very important for modularity was the work of Rudy Raff with Evo-Devo. Rudy Raff at Indiana in the last century and the early century, where for Evo-Devo, it's very important to have modules that can be composed, but it begs the question of multi-scale, especially temporal multi-scales, right? So it seems like a lot of what you're talking about is modularity at levels of biochemical reactions, genes. When you get up to Evo-Devo, then you talk, those have to be there, but also other components at higher scales and temporal scales. So the final question is, do you think the mathematical control theory that you're proposing has something to offer about this multi-scale aspects in time.
I mean, control theory was developed to be agnostic to what it was applied to. That was its whole, one of its objectives. So control theory is used in mechanical engineering. Artemis 2 is probably stuffed full of control theory stuff, right? Electrical engineering, of course, we heard pretty much across all engineering. So it's pretty agnostic. So I don't see why. But I mean, talking about beyond the cell, of course, there are some obvious real modules in the human body, which is, for example, organs. And the fact that we can transplant a pig heart into a human and it still works, that addresses the definition of a module, I think. So perhaps not, though, James.
Head your bet.
No, I was going to come back to the comment about Rudy's work because he was a truly great developmental biologist, evolutionary biologist, who never really worked closely with theorists, which was a pity. I tried to persuade him to for years. But one of his great discoveries was we usually think of evolution as working at the ends of sequences.
Yeah.
You add things downstream. You change the values of parameters.
Yeah.
And what Rudy showed was, at least in the case of sea urchins, that you had direct developing and indirect developing sea urchins, which had totally different developmental programs. And yet you could swap out the modules and they worked. So you could actually make what you'd consider to be a dramatic change early.
Yeah.
And the downstream effectors from that still worked, even though everything upstream was totally different.
Now, is that because the interfaces are the same? I mean, it doesn't matter how you implement it, right? So long as the function is the same.
Well, in one case, you start out with bilaterally symmetric organisms that swim around and look like little fish or tadpoles. In the other case, you start out with things that look like sea urchins. In one case, you go through metamorphosis. In the other case, you don't.
Yeah.
And yet you wind up with the same organism. He was asking a question evolutionarily. The obvious thing would have been that the direct developers were all descended from one and the indirect developers were descended from another. In fact, there were direct and indeveloper pairs that were evolutionarily close, and the choice between direct and indirect development happened again and again and again. And I think that's one that is certainly not a molecular scale question. It's a system scale question.
Yeah.
Okay, there will be.
That's it, I think, yeah.
There will be time after the next talk to discuss some more, but then in the interest of time, let's move on right into the next talk by Konstantin Mishakov from Rutgers.